Hello, everyone. Thank you very much for this introduction. I'm very happy to talk to you today about artificial axons, a novel biofidelic drug screening platform for neurological diseases. Neurodegeneration is a growing problem. There is nearly 1 billion people affected by some type of neurological disorders. These disorders often lead to neurodegeneration, where neurons die, leading to disabilities. Unfortunately, today we have no cures for many of these neurodegenerative diseases, including Alzheimer's, Parkinson's, and multiple sclerosis. Although these diseases are diverse, they have one feature in common. They often lead to damage of myelin. And myelin is this very important membrane that is wrapped by cells called oligodendrocytes around axons of neurons. Myelin works like an insulator, allowing neuron to conduct signals. And when myelin is damaged, neurons can't conduct anymore and die. Therefore, a key strategy in preventing neurodegeneration is stimulating myelin repair in the process called remyelination. Unfortunately, today we have no drugs available for remyelination. Part of the reason is that we also don't have adequate tools to study myelination and screen for remyelinating drugs. In order to do that, we need axon-like structure, a thin fiber with very low stiffness. So what is available today to study myelination? We have assays that are based on tissues and co-culture, which provide similar to in vivo environment, but they are too complex for drug screening. On the other end of the spectrum, we have tissue culture plates, which are very simple and reproducible, but they are flat and extremely stiff, therefore biologically irrelevant. Therefore, there is a need for three-dimensional solutions that will be biofidelic and at the same time reproducible and scalable. To fill that gap, we have engineered artificial axons. In this image, each fiber is made of a polymer and represents biological axon. It provides axon-like geometry and stiffness, and we can make those constructs in a very reproducible and scalable process. To create our solution, we have developed an advanced 3D printing technology. We provide axon-like geometry with micrometer scale diameters and stiffness below 1 kilopascal, matching that of biological axons. Our platform is reproducible and drug target agnostic. It can be used for many classes of drugs. It is also tunable. We can change the geometry and properties of the material, which allows us to model different diseases. We have demonstrated that artificial axons are biocompatible with neural cells, and you can observe directly myelin wrapping and quantify that process. As an example, you can see here green myelin membrane wrapped around artificial axon. The platform is scalable and compatible with standard laboratory practices, including live imaging of myelin wrapping. As the first commercial application, we focused on myelination assay because it is critical for development of therapies for neurodegeneration. There's also significant market in that sector, and there is no cures for remyelination. Artificial axons provide a better myelination assay. It is the only assay with quantifiable direct readout of myelin wrapping. You can see here in the image green wraps of myelin around artificial axons. You can quantify this process as a function of different drugs and doses, and our platform is capable of measuring those responses. Artificial axons are also cheaper and faster than current assays, and they are reproducible and suitable for high throughput applications. With recent advances in stem cell research, human-derived neural cells are becoming a standard in the development of neurological therapies. With access to individual patient cells, we are learning that neural system of each patient is unique. Therefore, personalized approaches to therapy development are needed. Artificial axons are ready for this next challenge. They are the only high-throughput myelination platform that is compatible with human cells. Using artificial axons, we are able to capture differences between patients in myelination, which opens opportunities for applications such as patient-optimized therapies or patient stratification in clinical trials. Key highlights of our technology include customization. We can generate any geometry and we can tune material properties, which allows us to model different diseases. The scalability, we can generate individual samples or the entire plate of samples. The high throughput, we can make that plate in minutes. And high resolution, we can generate micrometer scale features, providing high level of biofidelity in our models. With this versatile and flexible technology, we plan to expand interest of our future company beyond drug screening to disease models, drug disease mechanisms, biomarker discovery, and brain organoid research. Our platform provides a more predictive in vitro assay, which if used early in drug development process, it can reduce cost and time of drug development. We have validated our platform via strategic collaborations with multiple academic partners and also industrial collaborators. We plan to launch a company in the fall of 2021, and our business model relies on building drug discovery alliances with pharmaceutical partners and co-developing of therapeutics. We have been working on artificial axons for many years with a wonderful team of researchers with expertise in biomaterials and 3D printing and fantastic business advisors. I want to acknowledge our collaborators and funding and finish by inviting you to the Artificial Axon booth. We are launching in the fall of 2021 and are currently searching for CEO advisors, seed investment, partnerships with industry, and also passionate potential new team members. So I hope to see you at Artificial Axon booth. Thank you very much.